Hello, welcome to the lesson on drug discovery and clinical trials part 1. In this lesson, we will look at the overview of the drug discovery process, and we'll also look at various strategies in drug discovery as well as sources of potential drugs. Drug discovery is a very long but necessary process, and it involves many phases. Take a look at this diagram. Out of about 10,000 compounds that are initially discovered as potential drug candidates, only maybe 250 compounds will make it to the preclinical testing on animals. Following this, only maybe about 5 compounds will make it to clinical trials on humans, and perhaps only 1 compound will actually make it to the market. I also want to pause at this point and take a look at the different terms that we use. So you would have heard of pharmaceuticals or drugs or medicines, and these are different names, but they all mean the same thing. So a drug or medicinal product is any substance that would alter the physiological function of an organism, which is how the body works. And such chemicals would be used for the prevention, treatment, and alleviation of symptoms of a disease. The term medicine is sometimes used instead to distinguish therapeutic drugs from recreational drugs and other drugs such as opiates, which are used illegally. So with the understanding that drug refers to a medicine, let's take a closer look at the drug discovery process. As mentioned earlier, drug discovery is a very long, difficult, and expensive process. It takes about 10 to 15 years for a drug to move from the experimental stage in the lab to reach the actual patients. And the cost to discover a single drug could be in the hundreds of millions. Let's take a look at the whole process of drug discovery and development. First is the process of drug discovery, where potential drugs are discovered in the lab. Next, preclinical development is where these potential drugs are tested on lab-grown cells and animals before they are tested on humans. The company then has to submit an investigational new drug application or IND before starting clinical trials. The drug then goes through several phases of clinical trials. These are to test for safety, effectiveness, and to determine any side effects. If the drug is found to be safe and effective, the company submits a new drug application or NDA for FDA approval so that it can be marketed and sold in the US. So by the time the drug reaches the market, an average of 12 to 13 years would have elapsed since its first discovery, and the cost of research and development is estimated at about 2.6 billion. Let's take a closer look at some of the strategies used to discover new drugs. There are a variety of sources of potential drugs. Natural products have so far been the largest source of drugs. Existing drugs can also serve as templates for further chemical modification to improve their properties. New chemicals can be synthesized in the lab. In the past decade, computer-aided drug design has become a very useful tool in drug discovery. Biotechnology and genetic engineering has also risen to become a source of many new drugs today. Let's take a look at some examples of drugs from natural sources, which include animal, plant, microbial, or mineral origin. Did you know that insulin, which is used in the treatment of diabetes, was previously obtained from the pancreas of pigs and cows? The drug heparin, which is used to prevent blood clotting, was obtained from leeches. Insulin, as I mentioned previously, was obtained from pork pancreas. Thyroxine, the hormone, is obtained from the thyroid of animals. Vitamin B12 was actually obtained from liver extracts of animals. Cod liver oil obviously was obtained from codfish, and antitoxic sera, which are used to neutralize certain toxins, are produced in animals. So far, plants have been the largest source of the drugs that we have today. For example, morphine is obtained from the opium poppy and is a potent analgesic, which means it's a pain reliever. Codeine, which is a milder analgesic, is prepared from morphine. Reserpine was used for the treatment of high blood pressure. Quinine, an anti-malarial drug, was obtained from cinchona. And aspirin, which we all are very familiar with, was obtained from willow bark. You may have seen this flower in your very own garden. And did you know that vincristine and vinblastine, which are two anti-cancer drugs, were obtained from this Madagascar periwinkle plant? Another example of a drug obtained from natural source is digitoxin from Digitalis purpurea. This is called a cardiac glycoside, and it is used as a cardiotonic drug, which means that it has a strengthening effect on the heart. Another potential source of drugs is from microorganisms. I believe you are all familiar with the story of penicillin, which was discovered by accident by Dr. Alexander Fleming. After returning from a holiday, Dr. Fleming found some mold growing on a petri dish of bacteria. He noticed that the mold was able to kill the bacteria surrounding it and named the antibacterial substance penicillin. Besides penicillin, the antibiotic streptomycin is also from microorganism origin, and it was produced by the soil bacteria Streptomyces griseus. Besides living things in nature, drugs can also be found from mineral sources. For example, iodine is used as an antiseptic, and zinc oxide is used as a skin protectant. Today, biopharmaceuticals make up a large proportion of newly approved drugs. Biopharmaceuticals are protein drugs, for example, hormones and antibodies. These are manufactured using recombinant technology, where a gene of interest is isolated and then it is inserted into a vector, so it is recombined with the gene from another organism and it is cloned. Basically, it makes use of cells such as bacteria, yeast, or mammalian cells to synthesize these protein drugs. If you are interested in the basics of how recombinant technology works, you can refer to these two slides, which are inserted for your reference. Nowadays, computer softwares can be used to design new drugs. Computers can help scientists to look at 3D images of the drug molecules as well as their receptors, such as enzymes or DNA targets. Using this, it can predict whether the drug molecule would be able to interact well with the target. With so many compounds in the world, how do we discover compounds that may have a useful activity for a specific disease? With the use of automation, high-throughput screening can be carried out to screen millions of compounds to identify those that show activity, for example, against a certain disease protein. When a potential drug candidate is discovered, it is also characterized, meaning its physicochemical properties such as solubility, stability, or crystal structure are determined. These are important properties which affect their action in the body, also their ease of manufacturing, and their shelf life. The pharmaceutical company will also apply for a patent for the compound to protect their intellectual property. Patents can be filed based on novelty for new compounds or utility, such as a new use for an existing compound. Before a new drug is approved, it must first be shown to be safe and effective. and the manufacturers will have to provide proof of this by carrying out extensive testing. The different phases of testing would be described in part 2 of this lesson.